any of your other final exams. And if it does, you should let me know. So we have one more lab before the lab practical. So this Friday, we're going to be finishing up the muscles, and then we're also going to do the other sensations. So smell, specifically in hearing. And then we're going to have the lab practical on Friday, and that will be over the muscles, but also the sensory structure. So the eye and the ear, for example, are going to be a part of that lab practical. And then we have one more quiz, which will be next Monday, and it'll be over the information on the sensory system, okay, the special senses. So it'll be over the eye, and then what we're going to be talking about today. And then all of your mastering assignments are already online. And then I added this morning the extra credit assignment, which is due that Monday. You could do it after you take the final exam, but it's due Monday, that Monday of finals week at midnight. So you can upload that to the Canvas site. And remember that you have to have references so that I turn it in, will analyze your stuff and make sure that you haven't plagiarized. Okay, so that was a lot. So are there any questions about the final? Like, I can't believe that it's almost over. Although last weekend I was feeling like I thought it should already be over. Right? I was really sick last weekend. But. Okay, so we were talking about the eye. And one of the really interesting things I think about when I think about the eye is the way that lenses work is that they always flip the image. And so remember that the function of the lens is to focus the image on the back of the retina so that it can be clearly perceived. But just by the function of like physics, when you study, you took a physics class and you study lenses, one of the things you've learned is that lenses turn everything upside down. So all of the images that hit our retina are actually upside down. And so what that means is our brain then has to take that information and turn it back right side up so that we don't perceive everything upside down. Okay, so it's kind of an interesting effect of the inverted image here on the retina. And remember that the autonomic nervous system can affect the shape of the lens. And so here, the sympathetic activation actually causes the lens to become flatter so that we can see far away objects. And then the parasympathetic nervous system causes the lens to bulge so that we can see close up objects. And the ability to see things close up as they come near you tends to decrease with age. And that has to do with the lens becoming less plastic so that it actually doesn't have as much ability to change shape. And remember that that ability to change shape is called what? Accommodation, right? So you can accommodate looking at things clear or close and looking at things far away. So sometimes people are born with problems with the shape of their eyeball. So for example, if your eyeball is too long, then the image is going to fall in front of the retina. And if that convergence of those light rays are in the front of the retina, you'll notice that then the light scatters right here in the back. And so that would actually, right here, right? So that would actually cause the image to be blurry, right? And so then you can put a lens in front of the eye and change the way that the rays or the light is entering the eye, so that you get a fine focal point on the back of the retina, okay? So that's what happens in people that are like born or as they age, their eyes do not focus properly. And if your eyeball is too short, then the image falls behind the retina. So then you would put a different type of lens, a convex lens in front in order to focus the light rays on the back of the retina. Okay, so we talked about photoreceptors, and remember that photoreceptors in the eye come in two different types, the rods and the cones, which are named that because of their shape, and that these rods and the cones have in them specialized pigment, and that pigment is called rhodopsin. And remember that rhodopsin has two components, which is retinol and opsin. And opsin is the globular protein here. And so here you can see that this would be a three-dimensional protein that is highly folded, and it is also inserted into the membrane of a special type of organelle that is found, they're called rod discs that are, or cone discs, they're in cones. And then the retinol is a type of vitamin. Remember what type of vitamin is that? Vitamin A. Okay. So when light hits that, it causes retinol to disassociate from opsin, right? And that process is what is referred to as bleaching. So that then if all of your pigment has been turned into retinol and opsin, then you cannot see, and it takes a while for the reconversion, enzymes reconvert it so that you can get the pigment that can absorb the light energy. So remember that transduction in photoreceptors is a little bit odd in that rather than being depolarized when they're stimulated by light, they are actually depolarized in the dark. And so sodium ions just flood in, and the photoreceptor has a negative charge on the inside. So notice it's got a negative 40 millivolts, right? So that's depolarized. And it releases a neurotransmitter, which is inhibitory. And so you can see here that it causes IPSPs. And so that's a review. Remember we talked about those are inhibitory postsynaptic potentials. And so it keeps these other cells from firing and producing action potentials so that when it is in the light, the photoreceptor becomes hyperpolarized, the inhibitory neurotransmitters are not released, and so then therefore an action potential is produced. So it's just kind of exactly opposite or backwards from the other ways that transduction takes place in the other types of sensory receptors. Okay, you can bet that that's going to be on the quiz and the final exam. So that's why I was reviewing it. Okay. Okay, so transmission. So after transduction, transmission is how the action potential is sent into the central nervous system. So this is how the action potential is transmitted to the cerebral cortex, where it is consciously perceived. So remember that the optic nerve, that's an E, is one of the cranial nerves. Okay? And it is purely sensory, so it is conducting action potentials. And the optic nerve would then send the action potentials into the thalamus, because all sensory information goes through the thalamus, and then it can be transmitted to the visual cortex. And so if we looked at the pathway of this, right, one of the interesting things about our body is that oftentimes action potentials cross over, and they are actually sent to the opposite sides of the brain as where the perception for the sensory information originates. And so you'll notice that there's part of this that crosses over and part that doesn't. And so if you'll notice that kind of like in the peripheral vision right here, in the periphery of this eye, right, these action potentials cross over, and that crossing over is called the optic chiasma. So we saw that in our dissection of the sheep brain, right, very clearly, and also in the brain models, you can see the optic chiasma, and that's where they cross over, okay? So if I looked at the transmission, I would say that it goes from the photoreceptors in the eye, it goes to the next cells, which are called the bipolar cells, and then it goes to the ganglion cells. And then from the ganglion cells, it is transmitted to the optic nerve. So the optic nerve is actually the myelinated axons of the ganglion cells. And then from the optic nerve, it goes across the optic chiasma Okay, and then I'm going to go this way, and then it goes to the thalamus, and then it gets sent into the visual cortex. So sometimes people can have strokes, and when they have strokes on, say, for example, the left side of the brain, right here, that can affect their ability to see. Because if they have damage to the visual cortex, and sometimes when they get hit in the back of the head because the visual cortex is back here in the occipital lobe, sometimes that causes blindness as well, right? And that could also cause blindness in the opposite side or the opposite eye from where the damage occurs. So say my stroke maybe happens back here and I get damage to my right side of my visual cortex, it could actually cause me to lose vision in my left eye because of the way that it crosses over and is transmitted. Also remember that, I didn't put it here, but we have a primary visual cortex, and then we also have an association cortex, right? So we can see images, but we actually have to be able to make sense of those images. And so sometimes people get damage in their visual perception, where they can actually see an image, but they can't make sense of it. And I think I mentioned that before, where people can see somebody's face, but they can't recognize that face as being somebody that they know, right? They know it's a person's face, but they don't know it's their children's face, for example. Okay, so that's the transmission in the eye for vision, okay? So we're going to talk now about the other senses, and we're specifically going to talk about taste and smell and hearing. And taste and smell are associated with chemoreceptors. And specifically, chemoreceptors that are associated with taste buds, when we look at the taste buds, the majority of them are obviously located on the tongue, right? But there actually are some taste buds located on our cheeks. So tongue, a few on the inner cheek of the mouth, right? And also the pharynx. So it's not something that we think about too often, but you can, for somehow you can taste a little bit because there are some chemoreceptors located in these other areas of the oral cavity. When we look at these chemoreceptors, they detect chemicals in solution. And there's this old idea that you could actually map out different parts of the tongue where there were different types of receptors. So for example, it used to be thought that, like, for example, you could taste bitter in the back of the tongue, but sweet was more like in the center of the tongue. And so they created these tongue maps, but what they found is that those tongue maps are really deceiving, and that really there's not a distinct area where you typically taste more of one taste than the other. So those taste maps have kind of gone out of vogue in terms of the study of our ability to taste. So again, we have bitter, right? We have sweet. We have salty. We have sour. And then we also have umami. Does anybody remember what umami detects? Actually, spicy is different. Umami is savory. And so it is used to detect the presence of proteins. So things that are high in protein, like if you think about meat, for example, or even cheese. Cheese can taste very savory. That's umami. Okay. So bitter, just as a review, bitter is toxins, right? So a lot of plant toxins are bitter. Sweet would be energy, because those are simple carbohydrates that we can convert to ATP really quickly, right? Salty would be electrolytes. So like salt, right? So sodium and chloride and potassium are electrolytes that we need, ions, minerals. Sour is vitamin C, right? So we actually are dependent upon getting vitamin C in our food. So we can crave sour for vitamin C. And vitamin C is necessary for proper skin development, like you'll get sores on your skin if you're vitamin C deficient. So in the past, they called this scurvy, right? So pirates typically got lots of scurvy because they didn't have a source of vitamin C, right? And then umami is protein. Okay. Sometimes we put preservatives, or actually not preservatives, but flavor enhancers into our food to make them taste savory. And so a good example would be like chips, like Doritos, right? So what they put into Doritos, what do they put into Doritos to make them taste savory? Does anybody know? Powdered cheese, but there's actually something else. So have you heard of MSG?
Yeah.
Right. So MSG makes things taste really savory to us. So monosodium glutamate. Glutamate is actually an amino acid. So MSG makes us think that like Doritos have protein in them, but they don't really, and they're mainly carbohydrates and fats, right? But they make us think that they have protein, and then we eat them, and then we don't get protein, and then we kind of crave them, so it kind of makes us want to eat a lot of it, right? So MSG tends to make things very savory and kind of confuse our bodies.
How bad is it for you?
How bad is MSG? Well, there's lots of controversy over if it's bad. Some people have really negative reactions to it, and you probably heard of, it used to be that when you went to Chinese food places, for example, they use lots of MSG, right? So like those, noodles that people love, right? They have lots of MSG in them. They have lots of salt too. And those can be it's kind of addicting, right? And it's kind of gets you addicted to food that is not very good for you. Yep, that's the problem. Okay, the other thing I have to go on soda pop. Here's what I've learned about soda pop. Soda pop is full of salt. Why do they put it full of salt? Makes you thirsty. So if you're thirsty and you drink a soda pop, it is not going to quench your thirst because you are just taking in more electrolytes and it's going to make you more thirsty, right? And so you can actually get really dehydrated if you try to hydrate yourself on soda pop.
Gatorade do that,
Yeah, Gatorade's not very good for you. But if you are outside, like if you're a person that's working out outside and you're sweating a lot, you can actually become deficient in electrolytes. So then you need something like Gatorade. Or you can actually take salt pills.
On fire, they don't give us very much water.
Yeah, lots of sugar and lots of electrolytes.
But like if I'm sitting at home playing video games for 19 hours straight and I'm just drinking lots of Gatorade, probably not good for me.
No, that's, yeah. And actually, soda pop's got a lot more sodium in it than, I think probably more than Gatorade. It's got a lot of sodium in it, yeah.
One of my doctors told me that like when I'm dehydrated, to do a little bit of Gatorade and then dilute it with water.
Yes. Yes. So you dilute your Gatorade, yes. Yep. But it is true that you can become deficient in electrolytes if you're sweating a lot. Okay, so when we look at these taste receptors located on the tongue, okay, remember that we also have tonsils in the back of the tongue. And so when you get like the very back of your tongue starts to get swollen up, for example, this is the lingual tonsil, and that is due to an infection, right? Most of our taste buds are located in the middle part. They're called fungiform papillae. Don't need to know that name. And they actually have little pores in them. So if you looked at a taste bud underneath a microscope, you would see that there are receptor cells. Each taste bud has different types of receptor cells in it. And these receptor cells can bind different chemicals. And so chemicals come into solution. this receptor cell might be one that's going to bind salt, right? And so if it binds salt, then it's going to become depolarized and send an action potential out to the central, out through the nerves to the central nervous system. Okay? So these receptor cells are called gustatory cells. Right? And they have receptors that bind different chemicals, become depolarized, and transmit an action potential. So they would go to the gustatory cortex, right? So how you perceive the action potentials definitely depends upon just where they are being sent into the cerebral cortex so that you can consciously perceive that information. Okay. So this is a diagram that just shows this. And so there are, remember, cranial nerves that innervate the tongue. And so if we look at the major one, the facial nerve right here, right? The facial nerve innervates this, and so action potentials would be sent in to the central nervous system. They go up through the pons, up through the thalamus, remember, and then into the gustatory cortex. And the gustatory cortex is actually in that fifth lobe of the brain, which is called the insula, right? So instead of the occipital lobe, it's the insula lobe where that action potential gets sent. Okay. So let's look at smell. So smell is much more complicated. So in us, we have thousands of different odorant receptors. Oh, I did actually want to mention spicy. So instead of just being able to smell like 4 different things or 5 different things like we can taste, smell is much more fine-tuned. Dogs have like 10 times that amount. So they might have like 10,000 different odorant receptors. And then the fine-tuning of smell has to do with different receptors firing so that we can smell a complex smell. Like say, for example, the smell of an onion, for example, versus the taste of an onion. Okay. So I just wanted to mention spicy as a taste. Okay. This actually stimulates nociceptors. Excuse me, that's an N, N-O-C. And what are nociceptors? Pain, right? And so spicy actually stimulates pain receptors. And so that actually mimics the sensation of pain. And the chemical that does this is called capsaicin.
It's a chemical?
Yes, it's a chemical. And it is found in peppers, for example, hot peppers. And the reason I wanted to point this out is that you can go to Rite Aid or you can go into Safeway and they have like capsaicin patches, right? And you can actually put it on your skin and it stimulates pain receptors so that it produces endorphins, which reduce pain, right? So you can actually put capsaicin patches on different parts of your body. And people that have like oral cancer, oftentimes they're given capsaicin lozenges to suck on because that stimulation of those pain receptors then produces endorphins, and so it reduces their overall level of pain. Okay, yeah. So capsaicin is a chemical, and the thing about this is it does no damage, right? So it mimics damage to the tissue, but it actually doesn't cause any damage. And we have other things that kind of, this would be kind of hot burning We have other chemicals like peppermint, for example, that stimulates cold receptors and makes us feel like we're cold, right? But not pain receptors. Okay, so if we look at the sense of smell, so remember that we actually have olfactory receptors in the superior region of the nasal cavity. So superior means the top, right? And these actually dangle down through, they actually come through the cribriform plate of the skull. Okay, so they extend down through the cribriform, it's a C, cribriform plate of the ethmoid bone. And so these are specifically chemicals airborne, so these are odorants. So a lot of what we perceive as taste is actually coming from chemicals that get airborne, and then as we're eating them, then we smell them in, right? So like chocolate or like onions, they say that if you can't smell, you can't really even tell the difference sometimes between an onion and a apple, for example, because it's the smell that gives us a more defined, specific taste, what we call taste of those two different things. And so in your book, you have a picture of this structure. And so this is the cribriform plate, right? And notice that what sits right above the cribriform plate is the olfactory bulb. So the olfactory bulb is part of the unconscious part of our brain, but remember that it is also part of the limbic system. And so the limbic system is involved in emotional reflexes. And so smells are believed to be able to elicit emotional reflexes, maybe even unconscious emotional reflexes. So we don't even realize we're perceiving a particular chemical, but it can make us feel a particular way. Okay. So these are the olfactory receptors that are coming down, right? And so this is the mucus that is in your upper nasal passage, right? So this is very deep, right? So this is the single-celled, single layer of cells, the olfactory epithelial tissue, and then these receptors just kind of dangle down in here, right? And so chemicals would bind to them, cause an action potential to be produced, and then it gets sent into the olfactory bulb. One of the interesting things about your smell is you can actually damage these by using different chemicals. So for example, ammonia is like, well, it can make you pass out if you sniff it, right? But ammonia can actually also do damage to these receptors, right? So it can affect your sense of smell. So that these receptors actually are continually replaced after a period, I think it's like 60 days, they completely replace themselves. And so generally, if you do get damage to these receptors over time, your sense of smell will get better. But one of the problems in old people is that these receptors do not get repaired very frequently. And so old people tend to lose their sense of smell, and then they lose their desire to eat, right? Because like, when you think about eating, the pleasure that you get from taste is mainly smell, right? And so if nothing smells good to you, then you're not going to be hungry and you're not going to be interested in eating, right? So people sometimes just will stop eating and die because they have no desire to eat because it doesn't bring them pleasure in this sense. Okay, so if we look at the transmission, right? So we'll put that an odorant binds to receptor. And this is a direct transduction, right? So it causes sodium ion channels to open. Right? The receptor becomes depolarized. Right? It sends an action potential into the olfactory bulb. Okay. The olfactory bulb is limbic. It's also memory. So oftentimes when we talk about smells, they're based upon something that we remember. Like that smell reminds me of, right? So olfactory bulb, and then it goes to the olfactory tract. Because now we're in the central nervous system. And then it goes to the thalamus. And then it goes to the olfactory cortex, where we consciously perceive smell.
So how does it evoke smell? You know, like a certain smell.
It's because the olfactory bulb is connected to the hippocampus, and the hippocampus is part of the limbic system. And so once you've stimulated the olfactory bulb, then that can cause stimulation of the hippocampus and also memories. So like the smell of fireworks, for example, can bring back vivid memories. Okay? So it's all because they're interconnected via synapses between neurons. Okay, so in your book, they have a picture of this. Oops, didn't have that picture. Sorry. There is a picture in your book that shows how it goes into the brain and into the cortex. Okay. Last sensory. This is hearing. And we're actually also going to do equilibrium because hearing and equilibrium in us are interrelated. So when we look at hearing, this has to do with mechanoreceptors. And the stimulus are sound waves. So this is the stimulus. Okay. So I have a diagram that I'm going to give you that has the anatomy of the whole ear, and then on the other side it has the anatomy of the cochlea.
Thank you. Yes.
Make my quizzes pop.
No. Oh, you gave us an extra one.
Oh, I did.
So
Okay.
will be over how many lectures? Like all the lectures that we've done?
Sensory. So it's just over
So there's
ear,
one
the eye.
lecture that we didn't.
We didn't quiz over.
So it's not over today.
It's just going to be
Are
over.
you complaining that we didn't?
I'm not doing it. I just want to know what to study. So that was all
Yeah, like, we're fine with it.
But
It's just going to be over sensory. So it's just going to be over since we started this chapter on the sensory system. Okay, so we have mechanoreceptors. So sound waves are mechanical energy. And these mechanoreceptors, when they are physically deformed by the sound waves, then they generate an action potential. So it causes sodium ion channels to open and an action potential to be generated. So when we look at the ear structure itself, so we're going to look at the ear. Okay, we have the outer ear. The part of the ear which captures sound waves is known as the pinnae. Is there one N or two? I can't even remember on that one. Two? I just realized I can't remember. Do I have it? Oh, I don't have it. Okay, I think there's two. Is there two or one? Two, okay. So this is the outer ear. This captures sound waves. Some animals do not have an outer ear. So like fish, for example, don't have an outer ear, right? The sound waves just go through the water and then it goes right into their body. And then they have their mechanoreceptors located in the inside of their skull, right? But there's no outer ear. So terrestrial organisms have outer ears, and some of them have big ears. And so the bigger the ear, the more sound waves you're able to capture. So if you have bigger ears, if you want to hear better, right, you make your ears artificially bigger so that you can hear more, okay? Okay, so that's the outer ear. Then we have what is referred to as the auditory canal. And this transmits sound waves to the tympanic membrane.
This is in the outer ear.
This is still in the outer ear. Okay. This auditory canal has earwax in it, right? And so this is called cerumen, is earwax. And that has a protective feature. It's actually antimicrobial. So you can see how you can easily get infections, and sometimes you get what is called swimmer's ear, where the outer ear starts to itch really bad, right? And that is due to an infection, but the earwax actually helps to protect against infection. And earwax is really interesting because it's not produced really deep near the tympanic membrane, and it actually moves out by the movement of the epithelial cells as they migrate to the outside of the canal. And so if you push the cerumen too deep, sometimes it can get lodged against the tympanic membrane, and then you have to go in and the doctor actually has to clean your ears out, right? Because if you get that next to, there's no way to get that out. It doesn't tend to come out very easily. And some people produce a lot of earwax, and sometimes they have to go in and have them flushed out. They usually flush it out with warm water and it comes out. But typically it will come out on its own. Okay, so the tympanic membrane is kind of protected because it's not on the surface. Frogs, if you look at their ears, they actually have their tympanic membrane right on the surface of their skull. You can actually see it. But our tympanic membrane is protected. And it is composed of connective tissue. So this is the beginning of the middle ear. Okay. So it extends from the tympanic membrane to what is referred to as the oval window. Okay. So this is the middle part of the ear. And this middle part of the ear has ossicles. And these are ear bones. We actually have 3 ear bones. These are in order: malleus, and malleus means what? Hammer. Incus means anvil. So like if you were going to make a coin, right, you would strike the metal on the anvil, right? And then we have stapes, which means stirrup, right? But you need to know the more scientific, the malleus, the incus, and the stapes. So these are tiny, tiny little bones, and I actually have some in plastic, which we'll look at in lab. Tiny little bones, and they're connected to one another via joints. And so they actually have synovial joints located between these bones. So they're connected with synovial joints. Yes, they're tiny, tiny, tiny. We're gonna show you, I'm gonna show you a video. Okay. So what these do is that they amplify the sound. So this is the amplification of the sound waves. So it's gonna make them louder, bigger, right? So this is because we are terrestrial, and the sound waves travel really easily through air, But then when they get into our middle ear, then they have to go into the inner ear, which is filled with fluid. And so this is taking sound waves from air and it's making them bigger so that they will be strong enough to go into the fluid that is in our inner ear, okay? So this increases their strength for traveling from air into fluid of inner ear.
Is this all chapter 15? Or does it go over?
I think this is all chapter 15. Okay, so in the middle ear, we have a connection with the back of our throat. And so you probably have heard this referred to as the eustachian tube, right? Eustachian tube has another name. The eustachian tube is also called the pharyngotympanic tube. And the reason why it's called this is it goes from the pharynx to the middle ear, which has the tympanic membrane in it, right? So this eustachian tube connects our middle ear with our throat, and this allows for pressure equilibrium. So equilibrates pressure. So when your ears pop, right, we want the middle ear, which is filled with air, we want it not to like build up in pressure as we say, for example, go up to the top of a mountain. It needs to come into equilibrium, okay? So that's the eustachian tube. Okay, then this is where we have middle ear infections, right? So we can have the middle ear infection versus the outer ear infection, right? So this sometimes causes the tympanic membrane to rupture. Right, so it sometimes causes the tympanic membrane to rupture. So this is in children. If they constantly rupture, there's this idea that you could put tubes into the tympanic membrane to drain off that extra fluid. And this happens when your back of your throat gets, if you have a cold or infection, the back of your throat gets swollen up and it shuts off the pharyngotympanic tube, and then you get bacteria in the middle ear, and then you build up bad, like greenish or yellowish mucus in there, and then the eardrum will rupture. Okay, so this then connects to the inner ear, and in the inner ear, we have what is called the bony labyrinth. And this is where we have the semicircular canals and the vestibule plus the cochlea. So the cochlea, where we actually are able to sense sound, is in the inner bony labyrinth. That should be ia, cochlea. Okay, so on your diagram, we'll label these parts. Okay, so this would be the outer ear, the pinnae. I think it might be only one end. Can't remember. This right here is your auditory canal. Okay. This right here is my tympanic membrane. right? So this whole structure right here to the tympanic membrane is my outer ear. So notice that this would be the middle ear, and then the bony labyrinth, which is actually these little tiny canals through your skull bones. This is where these actually sit. Is the inner ear. So this would be the middle ear, and these are your bones. And so this first one that's connected to the tympanium, that is the malleus. The second one is the incus, and then the third one is the stapes. Now notice the shape of the stapes, right? It actually has an oval shape to it, and that is what we're going to call the oval window, right? Oh gosh, I'm losing it here. So this is my malleus. This is my incus. Get a cough drop. Okay. So when sound waves come in and they vibrate the tympanic membrane, then these bones amplify, and then the sound waves get transmitted into the inner ear via this structure right here, which is the oval window. Okay. There's another structure right here that's called the round window. It's probably really hard to see on your diagram, but it looks like this. This is my round window. And that is where sound waves leave the inner ear. So they go in through the oval and then out through the round window. Okay. These are my semicircular canals. Man, my pen's not working. Semicircular canals. This is my cochlea. Cochlea is the name of kind of like a shell. So the cochlea is a shell-like structure. And then the opening here, this is called the vestibule. So that area is called the vestibule. This would be my pharyngotympanic tube. Notice how narrow it is, right? So if that, you get bacteria in there, and this closes off because of an infection, then you can get an inner or middle ear infection. Inner ear infections are really rare because they would have to take place in this chamber right here. And so they are really devastating, specifically to equilibrium and to hearing. It's really bad to get inner ear infection. Okay, any questions about that so far?
Is the yellow the cochlear nerve?
Yeah, so this is the vestibular cochlear nerve. The vestibulocochlear nerve. Okay. So I'm going to show you the video, and then we'll talk about the back half of your page. So pay particular attention because it's going to show how sound waves move into the cochlea, and then how they stimulate the mechanoreceptors or the hair cells to then transmit an action potential out and be transmitted out through the vestibular cochlear nerve to the auditory cortex of the brain. Okay. So this is kind of a fun video because it shows kind of in great detail the movement of sound waves through the ear.
sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch or frequency produce a slower rate of vibration, and sounds of lower volume or amplitude produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spinal portion of the bony labyrinth known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spinal passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the 2 fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
Okay, let's turn the light back on. Okay, so let's look at your diagram. So if you turn that over, we see the structure right here. Okay, so this diagram right here shows the inside of the cochlea. And so the cochlea is filled with fluid. And it is part of the inner ear, and the cochlea is specifically responsible not for equilibrium, but for actually hearing sounds. And so if you look at a cross-section of the cochlea, there's these different chambers. And so the top chamber here is called a scala. So this is scala. And this one is the vestibuli. So sound waves come up through the fluid in the scala vestibuli, and then they leave via this other chamber right here, which is called the scala tympani. Okay, the middle one we could call scala media, or we'll just call it the cochlear duct. Okay. So the cochlear duct is actually where the receptor cells are located. And so if I look at this structure right here, this shows what is called the organ of corti. There's another name for it. Sometimes it's called the spiral organ. Okay, so if we look at these hair cells in here, these would be the receptors. So these are the hair cell receptors. So they would look something like this, where we would have a cell, and then on the surface we have cilia. And then we have an axon coming out. So what happens with the hair cells is if the hair cells bend, right, sodium ion channels open and sodium ions would flood in, and then this receptor is going to depolarize and an action potential is going to be generated. Okay. So this actually shows the picture of the little cilia on the hair cells inside of the organ of corti. So this is an electron microscope picture of that. Okay, so over the hair cells, we have what is called the tectoral membrane. And that is actually what vibrates and causes that vibration of the membrane, actually is what causes the hair cells to bend. Okay? And then underneath, kind of sandwiched between the tectoral membrane, this is actually called the basilar membrane. So the hair cells are sandwiched between two membranes, the basilar membrane and the tectoral membrane. So we need to talk a little bit about what sound is in terms of pitch and amplitude. So when we look at sound, right, we can look at the frequency of sound waves. So frequency just means the number of sound waves in a given period of time. And so if I look at in a graph here, if this is time on this axis, right? And this would be amplitude on this axis. We could have relatively high frequency sound, right? Versus a relatively low frequency sound, right? So this would be a higher frequency and this would be a lower frequency. So this is high frequency, low. Okay. That's the number of sound waves in a given period of time, right? So high pitch, lots of sound waves. Low pitch, a few sound waves, right? Then we can talk about its amplitude. The amplitude of the sound waves is the it's in the measurement of loudness, right? And this is measured in decibels. So how loud a sound is. And so this would be amplitude and time again. This would be a soft sound, right? And this would be a loud sound. Or soft or quiet. So those are kind of the two variables that change according to how we are hearing. So the way that the cochlea works is that the cochlea has hair cells that respond to different frequencies. So if you took the cochlea and you unwound it so that it was kind of like a straight tube, so it's a straight tube here, this would be called the base of the cochlea, and this is what is called the apex of the cochlea. And so at the base of the cochlea, we have higher frequency, so high pitch sounds. And these are shorter hairs. And at the apex we have a lower pitch, so low frequency sounds, and longer hairs. So it's very much like a musical instrument. So the sound waves come in, and if they're at a high pitch, then they generate action potentials in these receptors that are down here. If they're at a low pitch, they would generate action potentials down here. If they're loud, they're going to generate more action potentials. So an increase in amplitude generates more action potentials. So the sound is louder. So increase in amplitude generates more action potentials. Okay. Okay, so in your book, they actually have some really nice diagrams that demonstrate this. Okay, so this is change in frequency. The blue would be low frequency, the red would be high frequency. Change in amplitude, this would be loud sounds versus quiet sounds. And then if we look at the cochlea, and we unwind it, for example, the energy, remember, comes in via the scala vestibuli and out via the scala tympani. And it stimulates the different hair cells along the length of the cochlea. think there's actually a better picture here. Okay, so this would be a diagram from your book that shows that there are short, stiff fibers right here at the base and long, more floppy fibers at the apex. And so if we look at low frequency sounds, they're going to come in and they're going to stimulate receptors located near the apex, versus high frequency receptors or sound waves are going to stimulate those receptors that are near the base. And so this is the frequency hertz, right? 20,000 versus 20. Sometimes people have a hard time, specifically older males, sometimes have a hard time hearing high-pitched sounds, right? So you can actually get damage to the hair cells. Over time, we start to get damage and we start to get hearing loss, so that maybe we cannot hear certain frequencies. So high pitches are hard to hear, and maybe even low sounds, low pitches are hard to hear. Okay. And so that actually has to do with amplitude. So high amplitude sound waves can permanently deform hair cells, leading to hearing loss. So this is why it's important if you work with power tools or any other kind of machinery, right? You have to wear hearing protection, because you can get, if the sound is too loud, the hair cells will just lay down and then they will never pop back up again. And so then you have permanent hearing loss. And you can kind of tell when you've had some hearing damage, because if you've ever gone someplace and it's been so loud and then you walk out and you hear this, you know what I'm talking about, that fullness in your ears, that means that some of your hair cells are actually still bent over and they're not popping back up. And so that means that you've kind of already damaged some of your hair cells. So it's really important to protect your hearing so that you don't, because they can't be replaced once they have been damaged. Okay, so we'll talk more about, I'm going to talk about equilibrium on Monday. So we have a quiz on Monday, just over the last two lectures on chapter 15. Everybody got a review sheet? Final exam? Yep.
That's the best part. That's the best part.
See you later.
Have yet to repeat.
Yes. What time do you want to come in? I can.
My chair was broken.
Your chair. Oh, that. Some of them. Yeah. Okay. Thank you. What time do you want to come in tomorrow?
Didn't you have one at like 10?
I have, yes,
At 10:30?
10:30 to 11.
Okay,
Yeah.
I'll do that
Okay,
one.
okay.
Sounds good, thank you.
Hey.
Right, I know, I just think if I do it now, I have to pay it out.
Yeah, there's no
There's no standard class hasn't started yet, so you're good.
Thank you. I smart one. And smart one
So
too.
one to five.
Yeah, so three things. I said smart one, one, two.
The teacher will be talking right here.
I mean, you do.
I'm gonna do it like Snapchat. So it's pretty cool. When you go to university, you'll see a lot more of these classes. Yeah, just want to let you guys know. I had a biology class to do, and the instructor was asking questions, and some of the kids got their seats and laid it down on the desk. Yeah. Well, the instructor will either be in person or online. One of the chemistry teachers. There's one chemistry class, the other is online from, I think, the third one. So the front desk is the voice here, and they're seeing on the screen, they're doing around